Assalamu alaikum dear student, I am Dr. Sajad and this video is an introduction how to use the free online NCBI BLAST tool for your genomic research. So what is NCBI BLAST tool? BLAST stands for Basic Local Alignment Search Tool. It identifies similarity between biological sequences by comparing nucleotide or protein sequences to a database of sequences. इसमें हमारे पास एक क्वेरी सीक्वेंस होता है जो हमारा सीक्वेंस होता है जिसकी सिमिलरिटी हम चेक करना चाहते हैं डेटाबेस में अदर सीक्वेंसेस के साथ। तो इसमें हम एक क्वेरी सीक्वेंस को लेते हैं और उसको NCBI डेटाबेस में ब्लास्ट करते हैं टू चेक to get the result, the similarity result of our sequence with NCBI database. So first of all, you can open the NCBI database by writing NCBI in your Google and your Google Chrome and your Firefox, and then when you get this page, so you can click here the blast. So when you click the blast, it will you will get this blast page. Here you can see the nucleotide blast and protein blast, blast X, T blast N. We have already discussed these. What is blast X, what is blast P, what is blast N in our previous video. So if you are interested, you can watch that video as well. Here if you go down to this page, so there are some specialized searches, for example smart blast, primer blast, global align, CD search and LG blast. For example in smart blast you will find protein highly similar to your query sequence. Primer blast, you can design primer from your sequence and some other sequences. Global aligned, global alignment, and this one you can compare two sequences throughout their you can say across their entire span. It means that in this global alignment you can compare two sequences. For example, if one sequence is 1000 base pair, the other is also 1000, so it will compare the sequences from one base pair till 1000 base pair. And on the other hand, we have local alignment as well. So in local alignment you can compare some specific region of two sequences, but in global alignment you can compare the whole sequences. Okay, so this one we will click the nucleotide blast, and if you click the nucleotide blast you will come here in the query page. In the query page you can see here that there are multiple options: the blast N, blast P, blast X, T blast N, T blast X. So I will suggest you that you can check my video that what is blast N, what is blast P, what is blast X. Okay, the other how can we submit our query sequence to the NCBI database? So there are multiple ways, and we can submit our query sequence by multiple ways. The first way is to provide the accession number. So you can provide the accession number, so you can blast that sequence with the NCBI database. You can the second option, you can just paste the FASTA sequence, the nucleotide sequence in this query sequence portion. So you can, when you paste your FASTA sequence, you can blast your sequence through in the NCBI database. The third option, you can click here browse, and you can pick that your file from your computer, and you can upload to the NCBI database for the blast research. Here in this one you can see on the right side this query subbranch. So you can, if for example if your sequence is 1000 base pair, so you can select from 1 to 300 base pair, that I want to only blast from 1 base pair till 300 base pair in the NCBI database. So in this one you can select like the specify portion, a specific portion of your sequence to blast in NCBI database. Okay, and the other one, if you go down here, so you can see the choose the database, that there are multiple databases, there are many databases in NCBI. So which database you want to select for your blast. So if you see here, the default is core nucleotide core NT database. You can see here RepSeq select RNA sequence database. Here you can see nucleotide collection database, whole genome shotgun database. So you can select it based on your study and based on your requirement. But you can for the just for the identification and phylogenetic analysis, you should keep all these as default. If you want to know information about some database, you can click here, so it will give you information about that specific database, that what is whole genome shotgun database. So you can click here and it will give you information about that database. Okay, here we will keep the default setting, and you can also select here the RNA ITS database, genomic database. If you want to, like, if you want to minimize your search, if you want to narrow down your search in some specific database, then you can select these databases. In this one you can see here organism optional, so you can see this is the organism. If you want to search your sequence in some specific organism, you can write down the name. For example, if I want to search my sequence in some specific bacillus strain, bacillus bacteria, so I will mention that bacteria here. And if I want to exclude this bacteria from my blast, you can also click here and it will exclude from the blast result. Okay, you can add another organism, you can add another organism. So if you want to specify your sequence, if you have a sequence and you want to and there are some targeted organism in the database, and you want to only check, you compare your sequence with those organisms, you can specify those organism here. And if you want to exclude those organism, you can also exclude those organism here. Okay, so this is to exclude and include some specific organism in your blast search. Here you can see if you go down, so there the program selection. Here also some entries ID. If you want to get information about, you can click this question mark, and it will give you the it will get, you will get that page, the specific page. What is entries quality, what is like why I use this. So there are some specific like some condition in which you can use these type of options. So I will just remove this, and you can see here the program selection. In program selection, the default, you can select the default highly similar sequence, which is a mega blast, more dissimilar sequence, somewhat similar sequence, but I will select the highly similar sequence, which is mega blast. Here you can see in the blast, if you take it, it will show you the result, and it will open the result in other window. Like if you are using the Google Chrome, if you are using the Firefox, so it will not affect your this query page, and it will open in some other tab. It will open in another tab. So if you uncheck this, so then if you click the blast, so it will show result in this page. Okay, here you can see this plus sign. So if you click this and this plus sign, what it will do, here you can see maximum target sequences. Maximum target sequences, that how many sequence you want to include in your blast search. If you want to include only 10 sequence in your blast search, okay, you can select 50, you can select 100. So if you want to select how much sequences you want to select in your blast search, you can select here. You can also change the threshold expected threshold level. So but I will suggest that you should keep all these things as default. For example, if I paste this sequence here, you can see, and now I want to check that the result of this sequence, so I will click the blast, so it will open a new window. So here you can see the result. You can see on the left side that this portion is a summary portion. Here you can see the job title. What is your job title? You can see the RID, which is like you can see the accession number, like ID for this job, the program, which program you use for this. So we use BLAST10 because it was a nucleotide sequence. Which database we use core anti-nucleotide database. The query ID, which is your query ID. So this is my query ID. If you want to check your result later, so you can check by this query ID. The description, I didn't give any description. Molecular type, it was DNA nucleotide sequence, and the total length of my DNA is 1392 base pair. Okay. In this portion on the right side, you can see it is filter result. So you can filter your result by some specific organism. If you want to filter your result, you can write down some name here. For example, if you want to just filter the bacillus, there's some specific group of bacillus of bacteria, some specific species, some genus of bacillus or some other organism, you can write down it here. Okay, and you can add more than one organism here. You can filter your result based on the person identity. You can filter your result based on the e value, and you can filter your result based on the query coverage. So you can see the result here, that this is the result of our sequence we submitted. So here you can see that the first and this is the description of the sequence. This is the scientific name of that bacteria, and this is the maximum score of that blast. This is the total score of that blast, and this is the query coverage. What is the query coverage? The query coverage will show that how much percent it will cover by your sequence in the sequence in the database. For example, if you submit one sequence of 1000 base pair, so how much it is similar to the sequence in the database. If the database sequence is also 1000 base pair and both are 1000, so the query coverage will be 100%. But if your sequence length is smaller than that sequence in the NCBI database, so then the query coverage will be less. The e value. What is the e value? The e value, you can see that, for example, if the e value is 5, so it means that 5 hit. And this blast is like, you can say that accidentally, you can say that it is by chance. So 5 hit of this blast is by chance. So the more hit, the more incorrect result. If so the more the bigger value e value, it will give more error chances, and lower e value, it will give it will decrease the chances of error in your blast. So 0 is the perfect e value. You can see here the person identity. So how much similar your sequence is similar to the other sequence in the NCBI. So it show 84%. 84% my sequence is similar to the sequence in the NCBI database. You can see here the length of the sequence in the database. So you can see that my sequence is 16S ribosomal RNA sequence, but here you can see that most of the sequence are complete genome. So keep this in mind, that whenever you are trying to make a phylogenetic tree, you are trying to identify your organism by using 16S or ITS region. So then you have to select the 16S or ICS region in these organisms here, not the complete genome, because your sequence is definitely present in the complete genome, but the complete genome is more than 5 MB, 6 MB, and your sequence is only 300 base, 1300 or 1400 base pair. So it's better that you must select the 16S or ITS organism. Now how you will select those organism here? So you can see here that we can rearrange, we can ascending or descending on the basis of maximum score, total score, query coverage, e value, person identity, and I can also arrange on the sequence length. So if I click this accession length, so I will get the result. Here you can see that only one sequence with this 1400 base pair. So this sequence is 1400 base pair, and my sequence is 99% query coverage and 83% similar to this sequence. So now this is the description page. Now if you go to the graphical page, so the graphical page, you can see here that in graphical page you can see the alignment in a graphical format, and you can see here this is the alignment score. If there is a black line, so it means that the score is less than 40. If blue, then the score will be from 40 to 50, and if this is all in red, so it means the score is more than 200. Okay, so then on the description we discussed, the graphical summary we discussed. Now we will move to the alignment tab. So the alignment tab, you can see here your sequence comparison in your sequence alignment with the related sequence. So here you can see here this query sequence. The first one, it is our sequence which we submitted to NCBI for BLAST, and this is the subject's sequence. That is the sequence which is already present, already available in the NCBI database. Here you can see the score, and here you can see the identities that 1172 sequences out of 1397 sequences are similar. It means our sequence is 84% similar to the sequence in database, and there are 55 gap in our sequence and the sequence in the database. It means 3% gaps, okay, and both are plus strand. Now you can see here that here these lines show similarity, and you can see here this region, this is there is no line, so it means this is the difference. This is the difference between our sequence and the sequence in the database. In our sequence, the nucleotide is G, and the database, the nucleotide is A. Okay, so this is the difference. Now if you go down here, you can see here this is the gap, that in our sequence we have T and C, but in database sequence there is nothing, so it is a gap between the two sequences. So now you can see here that this is a gap between two sequences. If we go back to the description here, so you can see here that a distant tree. So whenever we are blasting sequence to the NCBI database, we are blasting sequence in the NCBI database to get about some information about some organism and to make the phylogenetic tree of that organism. So here if you click this distance tree, it will give you another you will get another page, and here you can see that it will give you it will show you a tree. So here in the tree you can see this this yellow color is our query sequence, and these are the sequences found in the NCBI database, our sequence. Here if you want to change your the name of the sequence, you can see here the taxonomic name of the sequence. You can click here and you can change your sequence name, the sequence name in the NCBI database. You can increase or decrease the tree by, you can zoom in, zoom out the tree, okay. And there are other tools. You can download this tree in different format. You can download this tree in ASN text file format, NIVIC format, Nexus format, PDF format. So for example, if you want to do some analysis by using the NCBI BLAST, you can make a phylogenetic tree inside NCBI database as well. This time I will blast a sequence by using the accession number in the NCBI database. So I already provide the accession number here. You can see here, and this accession number we will blast. So if we blast the accession number, so you will get the result here. This is the summary page about this sequence and about this query sequence submission to the NCBI database. All information here. You can see the filter result, and here you can see the description result. So first of all I want to sort out all these on the base of sequence length. So I will click here the accession length, and it will organize this in such way that it means that 1300 to 1400 base pair, all these have this 16S ribosomal RNA gene. So if you want to, this is select all sequences like 100 sequences, but I don't want to select 100 sequences. I want to select some specific sequences for my phylogenetic tree. Why I want to select these sequences? Because I want to make phylogenetic tree inside the NCBI database for you. So then I select almost these sequences. Then what you will do, you don't need to go here, because this is the other report, distance tree for the result. You can see here the distance tree of result. So when you click this one, so you will get to that page. So here you can see the tree from your selected sequences. Here this is the phylogenetic tree of your selected sequences. Here you can this one is your sequence, which is your query sequence, okay. And this is if you want to change the sequence title, only the taxonomic name. So if it will change it to the taxonomic name, and it will delete the other information. It will use only the species and the genus name. So you can you what you can do here, you can go to this tool and you can download this sequence in different formats. Next is format, which is you can visualize by different tree software. NIVIC format, PDF format. If I want to click the PDF format, so it will create a PDF, and it will download this tree in a PDF format. Okay. And if I want to, this is the PDF format tree, you can see here. If I want to check to download this tree in some NIVIC format, so I will click here and it download the tree in NIVIC format. And if I click here, you can see the NIVIC format. This is the NIVIC format. So I want to open this in Mega software, so you can open this in Mega software as well. Now this is the tree made by the we made this tree from the NCBI database. So I think this is the easy way for to make a phylogenetic tree to identify a species, or identify a bacteria or fungi or any microbe based on the NCBI database. So there are some different other options you can see here. The gene bank, if you want to get information about some specific accession number, so you click here the accession number, and this accession number, when you click this accession number, so it will give you information about that particular organism in the gene bank database and the gene bank file. Okay. And this is the MSA viewer. If you want to check this, so you can click here and it will give you more information about this about your sequence and the NCBI database. You can see here all the alignment, all anything. It is on plus strand, it is on minus strand, and this is or the gene type, the like ribosomal RNA, 16S ribosomal RNA gene. So it will give you more information about that. You can see, you can minimize here if you want only 10 sequences in your search here, so you can click here, it will show you only 10 sequences here, you can see here. Okay. If you want to 50 sequences, you can increase up to 50 sequence. If you select, if you want to delete a specific column, if you want to include some specific column, so you can do it from here. If you want to download the sequences, for example, if you want to make a phylogenetic tree but not in NCBI, if you want to make a phylogenetic tree, you can see here, I will select some specific sequences for the phylogenetic tree. So I will select it from here, and then I will go to this download option, and here you can download the sequences. If you want to download in these sequences in FASTA format, you can download here. If you want to download these sequences already aligned, so the NCBI will do also alignment for you, so you can download these sequences in align format as well. If you want to download the gene bank file of this, so you can download the gene bank. If I want to, for example, I want to just download in FASTA format, so I will click here in the FASTA format, and now you can see here these sequences are downloaded in FASTA format, and this is and already saved in my computer. So these are the sequences in FASTA format. So this was a brief video about the NCBI database for only those students who are beginner to this bioinformatics, and who only want to identify their bacteria, who want to make a phylogenetic tree by using their own sequences. So this video is only for the beginner. Thank you very much.